Hello everyone. I'll introduce the paper submitted to iMeta. The title is Microbial Synthetic Community ARC Prevents Aflatoxin and Increases Rhizobia-Legume Nodulation Couplingly. Legume crops face two global challenges in quality, yield, and safety. Peanut is a key crop for food, oil, and feed, but is vulnerable to aflatoxin, the most toxic and carcinogenic fungal toxin. Aflatoxin control has been a global priority since the 1960s. Legumes like peanut fix nitrogen, but field nodulation is sparse and inefficient. Improving nodulation, N2 fixation, and increasing yield has been an unresolved challenge for a century. N2 fixation demands high host energy, and balancing supernodulation with yield remains difficult. These two problems were studied independently. Since 2009, our team has monitored aflatoxin distribution and annual variation in Chinese peanuts. Analysis of 53 factors identified soil-borne Aspergillus as the main contamination source. Rhizosphere microbiome analysis revealed a seesaw pattern between Aspergillus and Rhizobia abundances in high and low-risk areas. As one increases, the other decreases. Therefore, we proposed a single technology to solve both problems simultaneously, biologically coupling soil source aflatoxin control with promotion of nodulation and nitrogen fixation. First, to screen for coupling bacteria, high and low-risk rhizosphere soils were analyzed. 20 genera negatively correlated with Aspergillus, 8 showed no negative correlation with Rhizobia and became candidate coupling genera. Using culturomics, thousands of isolates were obtained from peanut roots and soil. 127 strains from the 8 genera were identified. Co-culture with A. flavus yielded 7 highly inhibitory strains. 3 of them inhibited Rhizobia. Finally, 3 bacillus and 1 enterobacter were selected for SynCom assembly. Community assembly showed that the 4-strain SynCom had best inhibition of A. flavus. Field experiments further confirmed that this combination induced the highest number of nodules. This SynCom of 4 strains was named ARC. ARC alone could not induce nodulation, but when co-inoculated with Rhizobia, it significantly increased nodule number. ARC field trials from 2021 to 2024 at 325 sites across 4 major peanut regions in China showed that ARC significantly reduced surface mold spots and aflatoxigenic fungi, lowering aflatoxin levels by 85.6% compared to controls. Notably, ARC increased peanut nodules 5.3-fold and nitrogenase activity 7.4-fold. Active nodules normally decline late in growth, but ARC-treated plants retained abundant nodules at harvest, ensuring continuous nitrogen supply. Furthermore, ARC enhanced photosynthesis, plant growth, biomass, and yield. Third-party verified trials showed a stable yield increase of 19.8% across multiple years and sites. Overall, ARC achieved simultaneous aflatoxin control, supernodulation, high-efficiency N2 fixation, and yield increase in large-scale peanut production. Mechanistically, ARC restructured the microbiome. Enrichment analysis revealed an increase in nitrogen-fixing bacteria and a decrease in pathogenic fungi. ARC treatment downregulated 273 A. flavus genes. Go enrichment linked them to DNA repair, proteolysis, and fatty acid biosynthesis. When Rhizobium was co-cultured with ARC supernatant, colony number tripled, indicating ARC promoted rhizobial growth. Additionally, multiple nodulation genes in Rhizobia were induced. In ARC-treated peanut roots, 74 nodulation-related genes were upregulated, involved in early infection and nodule metabolism, indicating sustained nodulation and enhanced N2 fixation. Furthermore, ARC reprogrammed carbon dynamics, upregulating sweet sugar transporters in roots and nodules, thus enhancing carbon partitioning to roots. This created a powerful positive feedback loop, more carbon fueled N2 fixation, and N2 fixation further boosted plant growth and photosynthesis. In summary, we developed ARC, a dual-functional SynCom. In 4-year field trials across 325 sites, ARC reduced peanut aflatoxin by 85.6%, enhanced nodulation and nitrogenase activity, retained active nodules at harvest, and increased yield without the supernodulation penalty. Mechanistically, ARC inhibits multiple targets in A. flavus, recruits and activates nodulation/N2 fixation genes in Rhizobia and peanuts, and improves photosynthesis and carbon supply, balancing aflatoxin control, nodulation induction, and yield.